So actually, I'm going to just go ahead and get started. Okay, now I'm going to jump into actual things. Let's see if I can minimize this really quick. Sorry. Okay. So I've been here a long time. I've been able to see the kind of evolution of our long read systems since the very beginning. I started actually before we released the RS, so before we first commercially released any long read platform. But you can look at it historically and kind of look at the progression and how we've evolved over time in order to better meet our customer needs and really build a better product for our customers. Starting off back in 2010, 13, we had the RS and the RS2 system, and they ran on these smart cells that had either 75,000 or 150,000 ZMWs, so relatively low throughput, and I'd say exceptionally expensive at that point due to just the cost of runs and the low throughput. But from there, we kind of made this pretty large leap to the SQL system in 2015, where we released a new platform that had a much smaller footprint. The platform itself was much less expensive, so it was more accessible to a greater broader customer base. But we also released a new smart cell to go with it that had 1 million ZMWs. So the scale increased relatively dramatically pretty quickly, and therefore the cost per sample to customers. From there, we evolved that SQL system into a SQL2 system. The branding was not, I would say super creative at that point, but it got the point across that it was a new version of the SQL system. And the really, and then in October of 2020, released the SQL2E system. The real kind of innovation for the SQL2 systems was the 8 million ZMW smart cell, so 8x the amount of data per run relative to the SQL system, as well as on the E system, we built in onboard compute. So and also kind of released the new data type of HiFi data. So previously on the SQL system, we weren't generating HiFi data. We were generating what we called continuous long reads. So kind of like more similar, I'd say, to ONT that are noisy long reads. When we released the SQL2 system, we had the throughput enabled by the 8 million ZMWs to take our long reads and generate highly accurate long reads that we called HiFi reads. So we went one step up from what we were previously, along with the increased throughput. And when we built the E part, where we built the compute on board, it enabled customers to generate this HiFi data directly on the system, because there was an additional compute step to take these long reads and generate these HiFi long reads, highly accurate long reads. And with the SQL2 system, customers had to do it on their own, like HPC or their own analysis cluster or compute cluster. And with the 2E, all that work could be done on the system. It's a huge value prop, and it was a huge benefit to our customers. And then from there, in 2023, we shipped our first Revio systems. And I think the Revio system is kind of the state-of-the-art powerhouse right now of the PacBio long read platforms. And it took a huge leap forward. We went from 8 million ZMWs to 25 million, so about a threefold increase just in the density of ZMWs on the smart cell, as well as being able to sequence 4 smart cells at a time. So we had 4 independent stages, and I'll get into this a little bit later. 4 cells at a time, all within 24 hours. So we took a huge leap in terms of throughput and therefore cost to our samples, or sorry, per sample cost to our customers. And what this is really meant to show is that we've progressed a long way over time. And if you put your kind of future-facing glasses on, you can only imagine what's next, right? So taking a step back from our platforms, talked about HiFi a little bit. HiFi read being a highly accurate long read. What does a HiFi read look like? What is it? So I like this graphical representation, not graphical, but like figure representation that Jonas put together. It shows a approximate 20,000 base pair HiFi read, and these little arrows represent the errors. So this has a predicted accuracy of QV33, And these little red errors are the only errors within this highly accurate, nearly 20,000 base pair long read. So there's only 8 errors, and it comes out to about 99.96% accuracy. So this is what HiFi data can provide to our customers. Super long reads, super high accuracy. And our customers and our users are taking these HiFi reads, these highly accurate long reads, and are using them to transform genomics in the way we look at genomic research. The kind of value props of HiFi reads I have in the middle here, again, extraordinary accuracy, even coverage, which I think is a huge leg up when you look at like short read Illumina data. There's a ton of dips in coverage when you look at short read data, or specifically Illumina data. With HiFi reads, we don't have that same kind of sequence context bias, and we don't use amplification. They're all native reads or from native DNA. So we have really even coverage across entire genomes, which results in really complete genomes. And that's really critical when we're looking at especially human samples, where some of those drops in coverage may come from clinically relevant regions of the genome. HiFi reads allow our users to get down to allele resolution and really do some long-range phasing. And on top of it, we have the best performance for all variant classes when you look at single nucleotide variants, indels, structural variants relative to our competition. On the right-hand side is just a quick snapshot of looking at total errors across 3 different technologies. And this data was from Precision FDA Challenge that I've linked down here. So it's not data that we generated. It's kind of like a third-party benchmarking data set. But if you look at the amount of errors seen within these data sets across the different technologies, PacBio had by far the least amount, even relative to like Illumina, that had around 6x more errors, and especially lower than ONT, which had about a little more than 20 times as many errors relative to PacBio. So HiFi data really is the most accurate, complete data type out there on the market, and our customers are realizing that. And I wanted to say it's not just me saying that, it's not just the marketing person saying that. This is a quote from Molly Zeller at the University of Wisconsin, who's the core lab director at there. And she said, once there's no going back, it's HiFi or bust. So essentially, once you get the taste of HiFi, you need it. It really is that superior to the other data types. And that's the message that we want to get across to our customers. Some customers may say, there are other long read technologies out there that provide highly accurate long reads, but it's really not the case. The only highly accurate long read data type is PacBio HiFi reads. And if you look at a comparison of specifically ONT data across a couple of different processing varieties up here versus HiFi data, you'll notice there's a stark difference in the read accuracy distributions amongst those different technology types, where this green distribution up here that's pushed all the way to the right is HiFi reads, and this teal and purple distributions are ONT data sets, where they have a peak somewhere around 98 something percent accuracy, which is just under Q20. But really their distribution is so broad, so much of the data can't be used or is just thrown away or filtered away. When you look at HiFi reads, there are no data under Q20 or 99% accuracy, because a HiFi read is defined as being Q20 or 99% accuracy or greater. So you won't find anything less than 99% accurate reads in HiFi read data sets, which is why the distribution is pushed so much to the right. And our average is actually somewhere around Q30, so 99.9% accuracy. So not only do we have nothing less than 99% accuracy, the majority of our data actually comes from reads that are greater than 99% accurate. There's also Q20 plus chemistry that ONT likes to tout as being their high accuracy, one of their high accuracy chemistries. They have so many now. But even still, those data still fall short when compared to HiFi read accuracy. If you look at a similar read accuracy distribution, this isn't Fred's score, but we've put the percent accuracy below the kind of couple of the key points. When you overlay Q20 reads accuracy versus HiFi reads, again, you see like a mean or an average of around 99% accuracy with ONT reads, but the distribution is really broad. And half of the reads are going to come from reads that are less than 99% accurate or Q20 accuracy. And then when you look at HiFi, again, there are no reads below Q20 because by definition, a HiFi read is Q20 or greater. And the majority of our reads are coming from reads that are actually Q30 or higher in accuracy. So there is no comparison on quality. When you look at highly accurate long reads, really the leader is and will continue to be HiFi reads from PacBio. ONT also, this is a busy slide. I'm just going to focus on a couple of things. The other kind of challenge we get from ONT or ONT enthusiasts is that duplex reads can improve accuracy to be more competitive with HiFi reads. And it does do something, but it doesn't do enough. Ultimately, what it comes down to is that in the bottom right down here, if you look at the amount of errors produced by an 80-fold duplex assembly versus a 33-fold HiFi assembly, there's still almost 10 fewer errors in the HiFi assembly achieved with almost a third the coverage relative to the duplex data set. So there really is still, even with like all the bells and whistles OT can throw at it, there still is no comparison when it comes down to what is the most accurate long read data type. And then you can further drive home the point when you look at cost. All of this translates to how much is it going to cost me, and relative to any of the Promethean kind of configurations from ONT, Revio HiFi data is still going to be to have the lowest cost per gigabase. And the total throughput per cell is also going to be consistently higher on the Revio system relative to other ONT flow cells. If you all have questions, please stop me, or I will just keep talking for an hour and a half.
Yeah, Matt.
This is a question about. Oh, sorry, Chelsea.
I was going to say they'll unmute themselves. So there she goes. Go, Nicole.
Sorry, I thought Chelsea was talking. I thought I saw her mouth moving. Sorry. Yeah, I did have a question about the data where you showed the Q scores and like the accuracy with. Yeah, it was back a few, but yeah, this one. Illumina doesn't have a long read system though, right? So were you just overlaying the short read, or?
Yeah, that's
okay.
right.
Okay.
Yeah, and they do have. I'm spacing on what they call it, CLR reads or whatever.
Yeah.
But there's really, like, they've tried that a couple of different times in the past, and it's never panned out. And I think that the fact that we're hearing less and less about it after they announced it is good evidence that that's probably still the case. They still have to deal with all the things like amplification, cluster generation bias, all of those things that they have with short reads. They continue to have to deal with their artificial or computational long reads, whatever you want to call it. But yes, this specifically here is their short read data.
Okay, just want to make sure I wasn't missing something. Okay, thank you.
So again, I want to drive home the point, like, this isn't just your PacBio marketing team telling you PacBio is the best. If you look at benchmark data that's been published in peer-reviewed literature, they share the same sentiment, right? So a couple of quotes that I pulled out of this ABRF next-gen sequencing kind of benchmark study where they benchmark different platforms or different technologies amongst each other. All of these systems down here, so a pretty good pretty good set of systems or technologies. PacBio HiFi has the highest reference-based mapping rate and lowest non-mapping rate. Within both homopolymer and short tandem repeat classes, PacBio HiFi showed the lowest mismatch rate. And I think really what drives home the point is PacBio HiFi achieved the highest precision in assessing variant calls in clinically relevant regions. So again, it's not just us. You can pull from literature. You can pull from these benchmark studies that show really PacBio HiFi data is the really industry leader when it comes to accuracy. And you double it down with the read length, and it just makes it even that much more powerful. So what do HiFi reads enable for our customers? What can you do with them? You can almost do anything with HiFi reads. You can do similar or the exact same things you could do with other sequence technologies. Detect all variant types, detect epigenetics, highly accurate de novo assembly, phase variants in the haplotypes, sequence full-length transcripts, explore metagenomics in high resolution. If your customers have a sequencing need, HiFi data will have a solution for your customer. So I encourage you, as you talk to customers, it's easy to get kind of like pigeonholed into whole genome sequencing because it's the most common and most commonly used application. But try to explore other things that your customers are using sequence data for at their site or their account. And I guarantee you can find that they're using short reads for other things like RNA sequencing or 16S or metagenomic population screening or whatever, or other targeted sequencing, and see how you could convert those to HiFi data to use with PacBio HiFi data. And customers will also see the benefit, so it won't just be us. I'm going to skip that slide for now. Okay, that's HiFi reads. That's what they can provide and deliver to our customers, how they compare to some of our competitors and what they can be used for. Now, how do we generate PacBio long read or PacBio HiFi long reads? These are all of the on-market systems that we currently have right now that generate HiFi reads, or they can generate HiFi reads. The SQL system we no longer sell. It's been end of life. It hasn't been officially end of life yet, but it's the notifications that went out to customers, so I'm going to ignore that. I'm largely going to not speak to SQL 2. I'm going to touch on it, but really the main use case or the main generator of HiFi reads right now and going forward for PacBio is going to be on the Revio system. So I'm going to touch briefly the SQL 2 just so in case your customers ask about it or you have an interaction about it, you can speak to it and sound like you and know what you're talking about. So the SQL 2 system, kind of the footprint is very similar to the Revio system. It's a big kind of refrigerator box. The work deck looks very similar to the Revio, where you would load pipette tips or smart cells that come in a very similar form factor to the Revio. And then the sequencing plate and some other plastic reagents. It has a touch screen on the front right corner that also displays kind of some preliminary run stats, but really like a run progress. So it lets you know what cell it's on and what the status of that cell is, et cetera, and then the estimated time for completion. So not very complicated. Not a whole lot of stuff for the customer to have to worry about. But when we're looking at the SQL system, again, I'm going to say Revio all day. What's the difference between the SQLs, all the different SQL systems, and the Revio? The biggest or the largest differences probably come from the number of ZMWs that are on the system, the smart cells that are compatible with those systems. It goes from, again, 1 million to 8 million on the SQL 2, and then Revio 25 million. And then the other largest difference is probably the ability for the Revio system to sequence 4 smart cells in parallel, where all of our other systems, you can only sequence 1 smart cell at a time. Revio continues to have HiFi onboard HiFi generation, and only the SQL 2e system can do that, as well as onboard demultiplexing and onboard 5MC detection or HiFi kinetic detection, all kind of, which is kind of like a free benefit to detect 5MC with the SQL 2e and the Revio system. Our customers don't have to do any extra library prep. There's no additional modifications like bisulfite. We just use the sequencing kinetics. So basically how fast or slow we sequence around a given region of the genome of the sample. And we use that information to call modified bases. And this can all be done on the system for the Revio. And the sequel 2e can do it, and the ability to do this comes from just more powerful compute capabilities on the system. And I'll get into the compute on the Revio in a few slides. So before I move on to sequel 2, is there any, from sequel 2, are there any questions on any of the SQL systems? I like it, great. So our customers love HiFi long reads. What's holding them back from scaling with HiFi reads? This is kind of a problem with SQL mostly due to cost, somewhat due to throughput. So again, our request from customers in order to give them the ability to scale and sequence more and generate more HiFi reads is the cost, the throughput, and the ease of use. So those were kind of like the three main complaints, or I wouldn't say complaints, but like things that our customers wanted us to focus on in order to better improve our products. So we took those 3 points and used them as starting points to develop the Revio system. So we tried to drive down the cost per sample by increasing yields, decreasing runtimes, and also increasing throughput. We did that we increased yields through the 25 million ZMW SMRT Cell. We increased throughput by reducing or increasing the number of cells that we're able to sequence at a time from 1 to 4, and reduced the sequence time from 30 hours to 24 hours. And we made it more user-friendly or usable by reducing a lot of the plastics and kind of a few like things here and there, just to make the system more user-friendly and easier to run at scale, essentially. Key metrics, these are kind of things you should probably memorize in terms of the Revio system. The number of ZMWs per run is 100 million. So that's 4 25 million ZMW SMRT Cells can be run at a time. So you get 100 million. The yield is 90 gigabases of HiFi data per cell. So times 4 is 360 per run. Sequence time is 24. The recommended read length or insert size or library size is 15 to 20 KB. We have 5MC DNA methylation detection directly on the instrument with no additional library prep or other steps required. And then base quality, 90% of the bases that come off the Revio system will be Q30 or greater. This is all in our spec sheet also, if you want to look at it more in depth. The 3 kind of key applications for the Revio system are obviously going to be human genomes, because we can provide high throughput scale and reduced cost per genome. Single cell transcriptomes, we're able to really increase the number of transcripts that we look at per sample with the Revio throughput or yields. And then targeted gene panels, where you can sequence greater than 15,000 per year for even a large gene panel.
Matthew, before you continue, I was hoping to get clarification on what plastics were minimized in order to increase the ease of use.
I have a slide on that, so we will hold your question. I think that's okay. John, right?
Yeah.
Cool. Yeah. Sit tight, and I will get to that in, I think, 2 or 3 slides. So this is an image or picture of what the Revio looks like below the deck. So this is kind of like the brains, the guts of the Revio system. Up here, you can see this is the work deck. Customers are not exposed to this, so I added in a screenshot of this for those that aren't able to come on site and see what a Revio looks like. But each of these kind of units right here, these are the 4 independent stages and all of the equipment that is required to collect data from a SMRT Cell. So you can look at it as at a Revio system really as having 4 independent data collection modules that can be run completely independently and collect completely different samples. So you could envision, like, I'm a researcher I'm an oncology researcher. I have matched tissue pairs where a normal tissue sample and then a tumor tissue sample. And I also have RNA from the same sample that I want to collect isoseq data from using Kinnex. So you can run your normal sample here and get 90 gigs off this SMRT Cell. So you have 30-fold coverage of this sample. And then you can run 2 cells of a tumor, the tumor matched tumor sample. And typically researchers want to get a little bit higher coverage. So here they would, for the tumor sample, so they would get 30x coverage on each cell, combined to 60x coverage. And then from this SMRT Cell, they get all of their RNA data. So they can look at isoforms of this, this same sample, all within the same run, all within the same 24-hour period. So you can really look at it as like a project powerhouse instrument that can sequence multiple sample types all within the same run. So when we were designing the system based off of the customer feedback, we were aiming for these specific benefits, which we think we achieved. So there's scale, where we can sequence 1,300 human genomes per year, simplified ease of use. We have extraordinarily increased or improved compute power that uses Google Deep Consensus on the instrument and more, along with affordability down to $1,000 or $995 per human HiFi genome. And then this is how we achieved this relative to the Sequel II system. First, looking at scale, we have about 15x higher throughput or more throughput than the Sequel IIe system. And again, this is driven mostly by a 3x increase in the number of ZMWs per SMRT Cell, a 4x increase because we're able to sequence 4 SMRT Cells simultaneously, along with reduced run times, which allows our customers to churn out more samples over a given period of time. And all told, that comes out to 15x improvement. So we're looking at just under 90, 30x human HiFi genomes per year on the Sequel II system or Sequel IIe, up to 1,300 on the Revio system. So there's a huge benefit of running on the Revio system relative to Sequel II systems. And I don't have this here, but in terms of cost, it's about a 3.5 to 4x savings just on cost per genome alongside all of these other improvements. So it's not just that we're scaling, we're actually, with that scale, reducing the cost per genome to our customer. So I'm gonna hit on all the other kind of key benefits of the Revio system now. John, going back to your question, our consolidated consumables. So on the Sequel II, IIe systems, you need a sequencing plate, a sample plate. The sequencing plate is the reagent plate, basically. The sample plate is where the user would put their samples and put it on the instrument. You need a mixing plate, which would give a space for the instrument to mix the reagents and the sample on the instrument. A tube septa that goes with the SMRT Cell oil. So there's 5 different plastics. For all of these plastics, we combined all of them into a single plastic consumable for the Revio system, which is the Revio sequencing plate. So the sequencing plate now is used as a plate that contains the sequencing reagents. It's where the user loads the sample that they have. It's also used for mixing the reagents in the sample, and it contains the SMRT Cell oil. So all of those different things that were used on the Sequel II system are no longer necessary on the Revio system. It's just this one single plastic consumable. The second kind of ease of use benefit that the Revio has is we eliminated the need for external nitrogen. So John, I'm sure you know that Sequel II, Sequel systems, they need nitrogen in order, that's used for oxygen exclusion, which helps with things like read length and basically helping with polymerase survival. Previously on the Sequel systems, we would use nitrogen to displace that oxygen. But for the Revio, there's a new SMRT Cell design that's has basically an enclosure it's basically, I don't like using this word, but it's similar to a flow cell, where the surface of the SMRT Cell is covered up by this plastic layer here, and there are inlets and outlets here where we can input our sequencing reagents and sample, and then waste is dispensed through this. And when or is collected through this other waste channel. And then when the SMRT Cell goes to sequence, we layer on oil to these inlets and outlets in order to prevent oxygen from seeping in to the sequencing reaction. So we've achieved the same goal, but we no longer need nitrogen, which is a huge benefit to our customers who don't have to deal with building in a nitrogen line, if that's what they want to do, or dealing with nitrogen tanks and supply of those nitrogen tanks. And then the last thing, which I think I like the best out of the kind of simplified user experience and flexible workflows, is that the Revio system has the ability to, while sequencing, set up the work deck for the next, and queue the subsequent run. So within a 24-hour run, there's about 4 to 5 hours in the beginning of a run where you can't access the work deck and load your subsequent run. But after that, when all of the consumables that will be used for that run have been put under the work deck and onto the stages and been used essentially, the user can then go back to that instrument and preload their next run. So you can look at it as, if I set up a run on 10 a.m. on Monday for 4 SMRT cells, I can come back later that afternoon and set up my next 4 SMRT cell run that will kick off without me having to be there. What this allows for our customers is to have a seamless transition from run 1 to run 2 to run 3 and so on in order to best utilize their systems. Whereas previously they had to, and with the Sequel II system, and I think most other technologies, you have to wait for the run to completely finish before you set up your next run. And if the run time is, let's say, 30 hours, and you set up a run at 5 or 4 p.m. at the end of the day, your run is going to finish somewhere around midnight of the next day. And then the instrument's going to be sitting idle until you come into the workday, into the office the next workday in order to set up your next run. So we're basically allowing the customer to run on their schedule versus running on the instrument's schedule. So huge benefit to our customers, but it's also a benefit to us. If we can get our customers to utilize this feature, we can have them running more consistently and having less downtime, which then generates more consumable revenue, higher pull through per system. Okay, that was ease of use. We talked about scale, ease of use. Now talk about compute power. Relative to the Sequel II system, the Revio system has about 20x more compute. And what really drives this is we finally added GPUs to our system. So our servers that are on the Revio system use NVIDIA GPUs in order to process all of our data. We've also built in deep consensus from Google Health onto the instrument. And what this allows us to do is achieve higher accuracies with the same read lengths and same data collection, essentially. If you look at the bottom, there's this little diagram here where we show improved read accuracy on the Revio system that using deep consensus over Sequel II that doesn't use deep consensus. And Pat, you have a question?
Yeah, maybe we'll go over this in our next bioinformatics talk, but is deep consensus kind of inherent to HiFi? I don't know that I really understand what deep consensus is doing.
Yeah, sure. Let me try to explain that best I can. So deep consensus is essentially, I don't know, I think you all had Sunny's talk, but if you haven't yet, the way a HiFi read is generated is we have a circular molecule, right? And the polymerase sequences around that molecule many times. So we have reads, or you can look at them as sequence reads of the forward and reverse strand of a single molecule. And to generate a HiFi read, you take all of those passes around the molecule and stack them up and then build a consensus based off of the errors. And since our errors are random, that leads us to having low accuracy and no bias, because we don't have systematic bias. So they're random, and we basically build a consensus that can wash away those errors based off of coverage. What deep consensus does is it further improves that consensus building based off of machine learning that I can't technically speak to, but hopefully Paul can talk a little bit about that. It's basically another layer of consensus that uses Google Help's machine learning algorithm, Google Deep Consensus, in order to make that data more accurate. And actually, to achieve the same accuracy, you can use less data. So it's basically just a way of improving our accuracy and building a higher, more accurate consensus read or HiFi read with the same data type. Did that help at all?
Yeah. And it's go ahead.
Sorry, I didn't interrupt that. The other thing I was going to say is that it happens on every run. So there's no selection of whether or not you use it. It's inherent to all data that comes off the Revio system.
It's only on Revio, right? Not 2E?
Only on Revio, yeah.
Thanks, Matt.
So aside from the HiFi read generation, using deep consensus to help improve our HiFi read accuracy, we also do methylation calling, demultiplexing, and generate BAM files all on instrument. And if you look at other long read technologies, they really don't do much of this on instrument. It's all generated on off instrument, either on another server that the customer has to purchase in order to support their other platform, or in the cloud or somewhere else. But we basically spit this BAM file out off the system that contains all of these functions above. So we have a really more complete and rich data set coming directly off of our instrument at no additional cost to the customer. And then speaking of cost to customers, we've done a really good job of reducing our data file sizes. So previous format, so let's look at it as a SQL2 system format, we had about 1 byte per base in terms of file size that we required. We've reduced that by about half for the Revio system. And compared to ONT, it's about 20-fold lower. What does this mean for our customers? It means less costs go to data storage, less costs go to data analysis, and overall just general ease of use, having to work with file sizes that are much smaller. Another big kind of value prop that's lost, I think, sometimes when talking to long read customers. And then finally, affordability of the Revio system. This is kind of the last key benefit, but probably one that customers talk about the most, price. So we have, with the Revio system, we have about $1,000 30x human genome cost, and it comes from these 3 kits. So there's the Revio polymerase kit, which is what's used to prepare a sample for sequencing on the instrument, and then the Revio sequencing plate, which contains all of the plastics and reagents that we talked about previously, and then the Revio SMRT Cell tray. This is where the SMRT cells are held and put onto the system. So taking one reaction of each of these 3 consumables adds up to about $995 US list price per SMRT Cell, which is equivalent to a 30x whole genome, human whole genome. So again, the 4 key benefits of the Revio system, how we're trying to drive value to our customers, is through scale, being able to sequence about 1,300 human HiFi genomes per year, ease of use with simplified consumables, flexible run setup where the instrument runs on your schedule rather than it running on, or you running on its schedule, beefy compute power with NVIDIA GPUs and deep consensus and so much more analysis that we do on the instrument. And then finally, affordability, driving down the per genome cost of a HiFi genome with a Revio system. I didn't build in quizzes to this, so we can do kind of like a spot check or whatever, where you just can type in the answer to the chat. But so first kind of quiz question is, how many 30x human genomes can the Revio system produce per year? I can't see the chat either.
It's everyone is saying C.
C is the consensus. Yeah.
1,300.
Excellent. Good job. It is indeed C, 1,300 human genomes per year. You can really look at it right now as one SMRT Cell is equivalent to about a 30x human genome. And we can run about 1,300 SMRT Cells per year on the Revio system, therefore 1,300 30x human genomes.
What if you
What is
did
the
20x coverage? Just kidding.
Then you could do 50% more.
Okay.
So that scale could go up. So I mean, that's a good question. What do you do if your customer wants to run 20x genomes rather than 30-fold? Because 20x is going to be sufficient for most, if not all of our customer needs, aside from really like, I think RID and so rare inherited disease, and like telomere to telomere assemblies. So if a customer wants to sequence a 20x genome instead of a 30x, in order to maximize their value, they could barcode those samples. And let's say they have, just for ease of use, they have 2 human genomes they want 20x coverage for.
Mhm.
2 human genomes at 20x coverage would require 60 gigs of HiFi data per sample.
Mhm.
So they would need to run, actually, let's do 3 samples, 3 human genomes. They would need to run 2 cells, but they could barcode and pool those samples and run those pooled samples across 2 smart cells. Each smart cell would generate 90 gigs of data, so 180 gigs total, which would provide about 20x human genome coverage per sample. So there are ways that we can modify our workflow in order to best support and provide the best value for our customer depending on what their needs are.
Mhm. Okay. That's helpful. Thank you.
Yep. Okay, knowledge check number 2. Oh, sorry, go ahead.
I had a quick question. In terms of 30x coverage, is that like the kind of a standard amount of depth in order to go into the genome to have a reliable read?
So it depends on application, but I think 20 and 30x coverage are kind of like are kind of industry standards-ish, and a lot of technologies have built their messaging, et cetera, around 30x genomes. But it really depends on the use case of what our customers want to get out of it. And a lot of the times our customers can likely get away with 20x HiFi data because of all the things I've mentioned previously, the uniform coverage, lack of systematic bias, highly accurate reads. So I think 30-fold is kind of like a safe starting point. But as we talk to our customers, we can see if it's possible to drive down coverage requirements in order to save them money and be able to generate more genomes, given an X dollar amount that they have to spend.
I was just surprised that earlier when we were talking about Onzo, I remember them mentioning that it was like 3 to 8x is typical, 3 being the minimum and 8x being a typical depth.
So I'm not sure the context, but I would guess that it depends on the application that they're running. So I don't know the context, so I probably can't answer that. But I can. Who was that? Christy or Young who gave that talk?
I would have to search through my notes in order to find that.
That's okay. I can follow up and ask, and then write back in the chat what I hear back. Okay, knowledge check number 2. What is the US dollar list price for sequencing consumables per Revio smart cell? $7.95, $8.95, $9.95, or $11.95? John, do you mind being Nicole, my chat monitor?
Oh,
yeah,
I'm
I
here
think
too.
everyone's saying Steve.
Mommy, talk. No.
What's her answer?
She said C.
everyone
is for
said
cookie.
C.
C is for cookie. That is correct. C for cookie. Under $1,000 for a 30x genome at US list price. So I've talked about the Revio system, like the kind of key benefits, the kind of the value props, the cost. Now I'm going to speak briefly on the instrument design, so that if you ever speak to a customer or a prospect on it, you know what the different features of the system are. On the left is the Revio system, and I'm just going to kind of break down each of the external parts here. First is the status light up here. It has different colors based off of the state of the instrument. Green means it's idle and ready for sequencing. Blue means that it is currently sequencing or acquiring data. Orange means that there's an instrument issue, so contact tech support or PacBio tech support. And then white is instrument startup or upgrade or something like that. There's a status display window in the top right. It's not a touchscreen. It's just used for monitoring the instrument run. So you can see here, it actually shows the instrument name, when the door will be unlocked for the user to load the next run. And then it has the kind of smart cell progression here, moving from ready to prep to sequencing to done. And then how much time is associated with each of these runs. So each of these lines is a run here. This big black panel right here is the door. You press the button right here to open the door, and it slides up and down like this. Right here is just the logo light. It's a backlit PacBio logo that confirms the instrument is powered on. So when the instrument's off, this will be dark. On the right-hand side of the instrument below the status light, so like right here, is the power button that's in the groove of the panel. And then there's also an external USB port that's right below that you can see kind of zoomed up here. The work deck looks very similar to the SQL work deck that we looked at previously, but it's a little bit simplified because of the reduced consumables that are required for the run. So it's pretty basic. It's laid out pretty simple. All of these arrows make it look more complex than it actually is, but there's the reagent plates right here. These are two metal blocks that are cooled to 4 degrees. The 3 pipette tip locations that you can the user would add pipette tips to. There's 2 smart cell tray slots here. So the user can run 8 smart cells within a single run. The garbage is right here. This, or the trash that the instrument robot uses, is right there. This kind of gray metallic thing in the back is called the chip prep station. This is where the on-instrument robot or pipetter would put the smart cell. It's kind of like a prep station, really, like a chip prep station, where it will prep the smart cells for sequencing. So it loads the buffers, the samples, does mixing, things like that right here. There's a couple of instrument use things that are here. This is a tri-positioning sensor, or TPS. It just helps with the instrument confirming that where the pipette is, the pipette tip is relative to the pipetter on the instrument. And there are some auxiliary tip holders that are used by the FSEs, our field engineers. And then below here are the stages. So this trap door will lift up, and it will expose the 4 independent stages that we looked at previously. And so the user has no access to the stage. This trapdoor only opens during the run when the door is locked and closed. This is the pipetter. It acts as both a pipetter and a grabber, kind of like those video arcade games to grab the stuffed animals, except here we grab smart cells and pipette tips. And then there's a touch screen down at in the bottom right that the user will use to kind of initiate the sequencing run from. This is a close-up of the front display that I talked about earlier that you can look at your convenience. There's also a video of how to set up a sequencing run that we had one of our FASs record. So you can go back and look at that and see step-by-step how do you sequence or set up a sequencing run. Okay, now that's cool and everything. How does the Revio system perform though? So it performs really well. This is data that we had generated for release of the system. Release of a software update for the system previously, where we sequenced a number of different samples and sample types across human genomes, plant and animal genomes, all around 15 to 20 kb in size. The yields for one Revio smart cell were all, I think every one except one was 90 gigs or higher. The average was about 96. And the percent base quality equal to or greater than Q30 in every case was 90% or greater, because our spec is 90%. But on average, the base quality across all of these runs was about 91%. So the Revio is delivering on our spec and kind of what we say it can do. and it's able to achieve this kind of agnostic of sample type. This is just a few high-level metrics for a HE002 human sequencing run, where I wanted to show you so you can understand that read length and read quality, while high, are not a fixed amount. There will be a distribution. So this is a number of HiFi reads, and this is HiFi read length, so the size of a HiFi read. And the average is about 15 to, somewhere in the 15, 16 KB range. But there is a broad distribution from 10 KB all the way up to about 35 KB for this sample. And then this will be sample dependent, but you can expect that some reads will be slightly smaller or larger than that average HiFi read length. And then the same with read quality. While we have a mode that's maybe our median somewhere around Q30 or greater, there will be HiFi reads that have read qualities that are Q20, nothing less than Q20, because then it's not a HiFi read, but down to Q20, and some that are all the way up to Q50. And then this is the percent bases binned out by base quality. So again, the majority of our bases are going to be greater than Q30 in base quality. Same goes for methylation. Here we have IGV screenshots where we look at variant calling on HiFi reads relative to the Sequel II system, as well as 5MC calls, which are these the blue or red designations that designate hypo or hypermethylation. But essentially showing here that not only are they highly accurate, and here we're able to actually phase. These are the two different haplotypes within a sample, so we're able to phase the variance between haplotype. We're also able to do that with 5MC calls, where we can phase the different 5MC calls across the different haplotypes at a given locus. And again, that that 5MC calls, those are all done on instrument, no additional library prep, no additional sample modifications, all basically free data to the customer. And then looking at variant calling performance, other application-specific performance. If you remember earlier, I showed Precision FDA kind of benchmarking data of the HiFi data relative to other technologies. Here we show that the Revio system has equivalent or better variant calling performance relative to the Sequel II system. So we can say that everything we said previously holds true with the Revio, and it actually even gets a little bit better. And when we look at another commonly used application, genome assembly, the performance using those same data sets that I showed earlier of different human and plant and animal sets, we get really good contiguity, really exceptional genome assemblies from HiFi data that comes off of the Revio system. There are data sets available to share with your customers if they would like to further look at these, the data that we reviewed here. There are 5 data sets that I have listed here just to show basic performance of the kind of the metrics and specs that I spoke about previously. But we have a bunch of other data sets that are available for Revio HiFi data. Revio HiFi data sets available from various applications. From pure targeted sequencing, gene panels, plant and animal, MOS, or connects. Actually, I should put connects here, plus much more. And you can access these data sets publicly at pacbio.com/connect/datasets or pacbio.com/datasets. And you can share these with your customers, your prospects, to kind of give them real world examples of how HiFi data performs. So how is the how is the Revio system performing in customer hands? Our users are using Revio to basically scale and expand their research projects with HiFi data. They're generating more HiFi data, they have a faster ramp time, and there's, I shouldn't say preprints, there's many published articles that use data generated from the Revio system. I have this little screenshot or this little figure on the left-hand side that I really like to show how quickly our customers adopted and scaled with the Revio system. So we launched commercially back in the end of February, or started shipping more broadly end of February, beginning of March, like right here, of 2023. And by August of 2023, so roughly 6 months, about half a year after we've shipped our first Revio system, the Revio fleet yield had already surpassed the Sequel II fleet peak. So at any time across the Sequel II fleet history, the peak amount of data generated, the Revio was able to surpass that 6 months after launch. And I don't remember exactly how many systems were in the field at that point, but it was significantly less than there are now. So I think it goes to show that the usability improvements, the scale throughput improvements that we implemented in Revio are making a big difference, and our customers are able to adapt or adopt and scale and ramp with the Revio system much faster and easier than they have been in the past with Sequel II system. Anybody want to take, I didn't mention this, so anybody want to take a guess on which customer has achieved the highest one-day yields on the Revio in 2023? You can just spit out a guess because I can't see the chat.
Patrick says Radboud.
Good guess.
GeneDx.
Almost.
Ooh.
It was Earlham Institute. And they generated somewhere around 480 gigs of data in a single run. And this was, if you remember, our spec is 360 gigs in a run. So they exceeded it by 120 gigs. And I think what's great about it is that they tweeted it, and they kind of are being an advocate for us in the field and amongst other customers and prospects. So it's really great to see these kind of things where our customers are excelling and driving, pushing the limits of the Revio system. So we launched the Revio in 2023. Like I said, Q1 of 2023. We have improved upon it since then, and we will continue to improve upon the Revio as kind of our breadwinner for the long read systems, long read platforms we have at PacBio. In earlier this year, 9 months ago now, we released the Revio, what we called V13 or version 13 improvements. So it was really a software update, an instrument update to our system that we released broadly and recommended to all customers. And what this allowed us to do is to sequence more applications, even short fragments, all the way down to 1 KB in length, access more samples with new features like adaptive loading, run preview, higher accuracy, more consistent indel performance, more flexibility with movie time options, sustainability improvements to drive reliability, and better software. I'm going to kind of hit on some of these key things because I think they're important to talk about with our customers. The first is adaptive loading. Adaptive loading is a feature on the Revio system that allows the customers to achieve more consistent run performance. So as concentration of DNA on the SMRT Cell increases, our P1, or the amount of ZMWs that have been loaded with a DNA polymerase complex, also increases. However, if you increase too much, you get out of this ideal range and you get into this overloaded range, which impacts performance, basically, and the data that the customer receives in the end. What adaptive loading does, it acts as kind of like a backstop to prevent overloading. So it will, the instrument will monitor the loading on a SMRT Cell, and once it predicts that it will hit this ideal range, it terminates, like stops loading, and continues with data acquisition. So this way we can, irregardless of concentration, if it's too high, we can allow customers to hit more consistent, more predictable, and sometimes even higher yields than they previously have been. So it's a feature that was used a lot on the Sequel II system that the customers were asking for when we launched the Revio, that we finally got out into the field. And now I think it's somewhere around 90% of all runs use adaptive loading on the Revio system. So it's a really broadly adopted feature. We also have this run preview feature on the Revio system now with it was introduced in version 13 that gives a snapshot of data preview at 4 hours for a 24-hour run, at 4 hours after the run starts, and 23 hours. And what this allows our customers to do is to get some early feedback on run performance and better plan subsequent experiments. So you can imagine they look at the 4-hour preview and P1 loading, the percent of ZMWs loaded is really low. They can say, oh, I need to increase my loading concentration in order to hit a more optimal value here. And they can do that and set up the next run so that when this run stops, they already have their next run prepped and ready to go, versus waiting for the run to be fully done and have to wait to the run to be done in order to see these metrics and then plan the next experiment. So these previews provide things like the P1 loading, the HiFi read length estimate, a yield estimate at 23 hours, read quality estimate, but also barcode demux distributions. So it uses a portion of the data generated within a period of time from the sequencing run, and spits out basically the percent representation of each of the barcodes that it detected within a run. I really like this one because if you're multiplexing, let's just say 4 samples, and there's one sample that has a really low abundance or representation, then you know I need to sequence more of that sample, or I need to rebalance my pool or something like that. So it's a really nice feature or tool that our customers can use in order to better plan their subsequent experiments. Really quickly, version 13 allowed us to sequence multiple movie times from 12, 24, and 30. And the reason this is, I think, mostly common knowledge now, but the reason I put this in here is that based off of your customer's library size, there is an optimal acquisition time. So short fragments in the 1 to 5 KB range, we recommend a 12-hour movie or collection. 5 to 20 KB, we recommend 24 hours, and greater than 20 KB libraries, we would recommend using 30 hours. So there really is kind of a best match movie time for a given use case that your customers have. When you look at differences, so if you go back here and say, when I just want to run longer, even if I have shorter fragments in order to generate as much data as possible, it's up to the customer. They certainly can, but when we ran some samples internally, we don't see a huge gain in HiFi yields from 24 hours to 30 hours, but we see 110 to 113 gigs. But what we do see is improved read quality. So here we have Q30, average Q30 read quality in 24 hours, and in 30 hours it's Q33. So not huge, but 10% improvement in read quality can be significant depending on the application. So that, I think, should be the message to our customers, that if they have longer libraries and want to try to generate as high quality reads as possible, they can run 30 hours or maybe should run 30-hour movies. But if they don't really have the need for it, it's kind of just excess at that point. Okay, that's technology, that's the system, that's performance. I'm running out of time, so I will quickly go through some promotions and other things relevant to kind of the commercial side of the Revio system. So system pricing, or Revio pricing, we have list price of US dollars is $779,000 for the instrument, and then $995 per genome, which breaks down for one reaction per those 3 kits that I mentioned earlier. We do have a couple of different Revio system promotions that we're running right now. The 779 CapEx can be prohibitive for some of our customers. So we have 2 promotions that we call Run Revio promotions that significantly reduce that CapEx upfront requirement. One by half, so about $395K CapEx, with a consumable commitment of 384 smart cells at a slightly higher list price, so $1,400 each rather than that $995. that we would require the customer to receive and order and receive within a year after installation. So it's essentially a way for if a customer has like OpEx budget, but they don't have as much CapEx budget as they need, they can use their limited CapEx budget and their excess OpEx budget in order to bring the Revio in-house. We also have a Run Revio that's a $0 CapEx. So this is similar to the $395 CapEx, but $0 upfront. Just a requirement for another 384 Smart Cell consumable requirement at a little bit higher list price or price each. So 2,500 per Smart Cell rather than that 1,000. So again, it's a way if customers have a grant that they can only use for OpEx, they can get a Revio in-house and fund it through their OpEx grant via the consumable commitment of this promotion. These expire end of this year. Customer needs to accept installation. The PO needs to be put through by the 16th of December. Customer needs to accept the instrument by the 20th of December. There are multi-unit promotions that you can reference, but essentially the more units customers buy, the higher the discount rate is going to be for the Revio system.
Matt, do we recognize revenue at install or at shipment?
We recognize it at install.
Install, okay.
Yeah. So the customer basically for those promotions, the customer has to have it not just accept it, but like install it by this one. Yeah.
Okay. So those deals would already be in the system. Yeah. Okay.
We have a couple instrument bundles, a Revio and a Sequel II bundle, and a Revio and an Onso bundle, where customers can get the best of both worlds at a pretty significant discount. I don't have time to cover the business case analysis, but I'm just going to show the slides. There are a number of different kind of comparisons that we could make from the Sequel II to the Revio system, or just kind of operating expenses of the Revio system at a given customer account. You can find these slides here, the Revio system business case analysis slides in Seismic. There's a link here. If you haven't explored Seismic, you should. There's a number of kind of comparisons and looking at sequencing consumable costs by application and system, showing essentially the cost benefits of a Revio system versus a Sequel II system for a couple of different project types. There's a sequencing consumables cost versus outsourcing estimate. So if you have a customer that's on the fence and they might want to just outsource samples versus bringing the technology in-house, you can look at how much samples would cost to sequence in-house versus estimated markups at a service provider. And then finally, kind of like a break-even period estimation based off of utilization of a Revio system. So at a given, this is more for like a core lab or a service provider, at a given markup, X markup relative to list and a given utilization, how many months would it take to break even for the purchase of a Revio system? So driving home these four key benefits of the Revio system: scale, ease of use, compute power, and affordability, really driving home the value of HiFi long reads for our customers. There are a number of system assets that you have access to. There are demo instruments for like field marketing. I'm sure you'll be exposed to those that we use a lot at conferences and other events. There's the PacBio.com Revio page that has basically all of the Revio relevant information. There are Revio data sets that we have available to share to customers, as well as Seismic and other kind of social tools that we have. Go check out Seismic. Seismic, I don't know if I have the link in here, but on Seismic, there's a Revio landing page that has all of Revio collateral information, trainings, those business case slides that I went over, customer-facing decks, internal-only FAQs, sole source, grant support documents. There's a number of, it's a huge resource. So please go check out the RIF